Welcome, everybody. Great pleasure to welcome you to the second in the series of the Association of Physicians webinar series in Emerging Issues in Medicine. My name is Colin Dayan from Cardiff University, and my co-chair is Seamas Donnelly from Trinity College, Dublin. We have a fantastic lineup of speakers for you this evening, and the format as we've had in our previous webinars, we're going to ask them all to just talk for 5 or 6 minutes to introduce their perspective on the topic tonight, which is the COVID Enigma, and then we'll enter into a discussion to try and get to the bottom of this emerging challenge. I'm going to pass over now to Seamas to introduce our speakers.
Thank you very much, Colin. As we have 4 very eminent speakers from around the world, our first speaker this evening will be Professor Gareth Fitzgerald, and Gareth is the Director of the Institute for Translational Medicine and Therapeutics at the Perelman School of Medicine at the University of Pennsylvania. That'll be followed by my Trinity College Dublin colleague, Professor Luke O'Neill, who's Professor of Biochemistry. And then we've Rick Buchanan, who's Professor of Medicine and Chief Rheumatology at Yale University. And then our fourth speaker will be Timothy Tellinghuisen, who's Head of Virology at Roche, based in Basel in Switzerland. And as Colin has mentioned, the speakers will be short, sharp and focused. And the main purpose of the webinar is to instill a very vibrant question and answer session for the majority of the 60 minutes. And in that context, if you wish to ask questions, we're going to use the chat format. So just type in your questions and we will address them due to the Q&A format after our speakers. I unfortunately my camera isn't working, so I'm just without video and you just hear me by sound. So without further ado, we'll proceed to our first speaker, Professor Gareth Fitzgerald. And Claire, if you could load up Gareth's slide set.
Okay. So, can everybody hear me? Hope so.
Yes.
Thanks. So, good afternoon from Philadelphia. As you can see, we are entering our second wave. These are the admissions to our health system. I thought I'd frame the conversation, as I have to speak first, by illustrating a few things that we don't understand. The remarkable clinical heterogeneity of the infected phenotype, the long tail of COVID-19, reinfection, it seems to occur. What does it mean? And the strength, persistence of post-infection immunity, and indeed immunity resulting from vaccination, and in particular the pediatric response to COVID-19. What we do know is masks reduce the chance that you will infect others and probably also protect you, that social distancing works and that hand washing reduces transmission. And a bit more speculatively, that we can suppress community transmission by closing other sites to protect the likelihood of schools remaining open, that mouthwashes reduce transmission and infective load, and that there seems to be little risk of fomite-based transmission. So one of the things that has been very striking in this pandemic has been the diverse impact it has on various ethnic groups. And that's illustrated in this slide, where we were looking at antibodies evoked by the virus in infected patients, particularly those presenting in pregnancy. But you can see at the bottom of the slide that we saw this remarkable contrast in the incidence of antibodies in Black and Hispanic or Latino women presenting in pregnancy compared to white women or Asian women. And obviously there are many coincident factors with ethnicity, not least of them being socioeconomic, but understanding the basis of the ethnic diversity and susceptibility to infection is a topic that we and many others are interested in as well. So I thought I'd talk a little bit briefly about what we have learned. We've learned the value of old-style randomized control trials, exemplified by the recovery platform that has delivered to us, dexamethasone, efficacious if you're very sick, maybe even harmful if you're not very sick, and a clear answer as far as hydroxychloroquine is concerned. We've seen also the evolution of information over time, as illustrated by remdesivir, a bit of evidence suggesting its efficacy that led to its FDA approval in this country, a more clear-cut answer in the negative from WHO. We have some suggestions from the anti-IL-6 world that particularly with interventions that are relevant to the immunological response to the virus, the timing may be very important. We've got recent evidence from tocilizumab from the Imperial group that suggests efficacy in contrast to some earlier studies, and timing may have been one of the variables that contrast those answers. In terms of antibody-directed therapeutics, we have emerging evidence as far as that's concerned. And then we have the adaptation of trial designs that were applied, particularly in the cancer field, to look for combinatorial efficacy, particularly at the level of phase 2, adapted to asking the same sorts of questions with respect to the pandemic. In the last couple of slides, I'd just like to illustrate a couple of things. Science is beginning to deliver us some answers with respect to this issue of clinical heterogeneity. For me, a remarkable couple of papers came from the Casanova group in Rockefeller recently, initially identifying inborn errors in genes in the type 1 interferon pathway relating to the severity of the disease phenotype. And then the remarkable discovery of the presence of autoantibodies against these type 1 interferons in patients with the severe phenotype. I suspect that this is just one of the pathways that may be represented in this respect that will illuminate our understanding of the clinical heterogeneity of the pandemic. We're particularly interested in the role of the complement pathway and its activation as an explanation for not just the diverse organ damage that one has seen in this disease, but also the hemostatic activation and the thrombotic phenotypes that are concerned. We're particularly interested also in the role that lipids may play here. We know that lipids modulate viral entry, viral replication, but also play potentially very important roles in modulating the inflammatory response to the disease. And through the work of John Querry, this slide only serves to indicate that with deep immunophenotyping of patients with this disease, there are remarkable, clear, distinct immunophenotypes, even in patients presenting with an apparently homogeneous severe clinical phenotype. So finally, the approach that we've taken based on that is to, first of all, develop our capacity for high throughput testing, to apply that to widely in the population, to integrate that with tracking approaches and telehealth so that we can look at people with mild disease who are at home and to develop an infrastructure that allows for us to collect biosamples at that early stage of the disease. As patients evolve in the disease and present to hospital, we have a pipeline that integrates genetics with multi-omics analysis and complement, as I've mentioned, integrated with deep immunophenotyping. We've modified our electronic health record to allow that to be integrated with this type of information, but of course, detailed clinical phenotyping requires abstraction, and we're doing that as well. And very importantly, this type of approach, we've developed a single overarching consent that also covers follow-up when the patients are discharged. And finally, our focus institutionally has been on observational studies of the type that focus on explanations that might be relevant to disease heterogeneity. We've made foundational contributions to RNA vaccinology through the work of Drew Weissman and his colleagues, to DNA vaccinology through the work of Dave Weiner and his colleagues, and therefore are very involved in those types of trials. But for other therapeutic entities, we have really confined our focus to participation in very large-scale trials that have the power to deliver an answer, and exploratory proof of concept trials driven by local research. And examples are in the complement field, where both John Lambros and Wenxia Song have made foundational contributions to our understanding of complement activation in vivo. With that, I'll conclude and stop sharing and clear the screen.
Thank you very much, Garrett. We'll proceed to the second speaker, which is Luke O'Neill. He's going to discuss potential novel therapies, particularly in relation to the cytokine storm in COVID-19. And as we get his slide set ready, just to remind participants that any questions for the panelists and the speakers, use the chat option at the bottom of your screen.
Thank you, Seamus. Here are my slides, so we can see them. I'm going to talk about the cytokine storm. I don't think it's a storm. It might be a bit of drizzle with this disease. We can discuss that point in a minute, I guess. But my own lab has got 3 projects running on COVID-19. I work mainly on inflammation and inflammatory diseases. I've also worked on LPS for donkey's years, which could be relevant, of course, to this disease as well. But back in February, March, we switched over to working on COVID for obvious reasons. And I'm going to give you 2 bits of data, actually, that we just got in the last month or so. A couple of things to touch on now. I guess Garrett again referred to some of these things. I guess the big debate, is it worse than flu? Of course it is. Here's some data for you, which I thought I'd show. These are all these COVID-19 studies and the infection fatality rate versus seasonal influenza. And you can see past the age of 40, you're getting a 13-fold increased risk of death from this disease. So that debate should end, in my opinion. This is not like the flu, which is a big thing that's debated in Ireland the whole time. Another quick thing is we work on BCG a lot in my lab. With Mihai Netea, we did stuff on epigenetic reprogramming on monocytes. This will drive cytokines, prime gene expression. There's now 2 big trials running with BCG against COVID-19. It was shown to protect against influenza and RSV, for instance, and now the big question is, will it protect against COVID too? This is like innate immune memory, I guess, and that's something we might come back to. It involves cytokines, so I put it in there as an example. Clearly, it's an inflammatory condition, as you all know. Macrophages, lymphocytes, cytokines, everything you want to see are there. And this is a recent JCI paper, I think it could be relevant to us. What's it like compared to ARDS and sepsis? And it seems to be very similar. And these are all these various cytokine markers here with no real difference if you look at the totality of all the data between COVID, ARDS and sepsis. And in this paper, I thought there was a good sentence in the discussion: broad use of immunosuppressive therapies in ARDS has failed in numerous phase 3 studies. Use of these therapies in unaffected patients with COVID-19 may be unwarranted. Now remember, people have spent 50 years trying to handle LPS and sepsis and these things and failed. And the big question is, what makes this disease different? Why do we think blocking cytokines might work, given all the failures over the years with this? Now, this is an overview. It came out in Science, I think about maybe 4 months ago now, of the various approaches. Remdesivir doesn't work. Let's start with that. Here's the inflammatory cytokines. Many targets here. GM-CSF is very interesting, for instance. Tocilizumab as well. Chloroquine is gone, and there's the antibody-based therapies. Obviously, it's getting better in hospitals. We all know this now. The survival rate's gone up a lot. This is a good study that was in the New England Journal. You know, comparing April to May or late June and the different ages, there's clear survival happening. The big question is, why is this? Is it therapy? Is it proning? All these various things you'll all be aware of. Is it dexamethasone? Garrett touched on this. Now, many of us worked on dexamethasone. Part of my PhD was dexamethasone, and I was surprised when it came back in this way. Clear efficacy, though you all know this data, I guess. Probably 10 to 15% benefit overall, you'd say, if we take all the studies combined. But clearly, it's making a difference in this more late-stage disease. Now, finally, my own work, the NLRP3 inflammasome, my lab is obsessed with this pathway. Lots of stuff implicating this in COVID, driving IL-1, IL-18, and pyroptosis, those 3 outputs from this. This is a recent review. I just cribbed this slide from driving the so-called cytokine storm. Good rationale here, we think. Our own data, we're measuring cytokines in clinical samples from St. James's Hospital with Niall Conlon. IL-18 stands out as a biomarker. Other labs have shown this as well. Here's severe patients compared to non-severe. IL-18 clearly stands up. So it does seem to involve NLRP3. macrophage activation syndrome type scenario, and many labs are showing this, and that's of interest clearly clinically. You never know, these could be targets. However, the canakinumab trial failed. Novartis tried to block IL-1 beta in COVID-19 and got no effect. Tocilizumab, it's still, the jury's out. There was a failure. There's been some successes. That's blocking IL-6, as we all know. I don't know. I mean, as Gareth said, it could be a timing issue. Maybe it's not going to be easy to block these cytokines and see clinical impact, I think. There are 2 trials running at the moment with NLRP3 inhibitors. Novartis and Olitech are running those trials at the moment. I guess we'll see what will happen. Remember, NLRP3 will also drive cell death. It's not just IL-1, and will drive IL-18. So maybe they're more important than IL-1. That could be why canakinumab might have failed, or it could have been the timing issue, as we said. Now, finally, a bit of my own work. We're obsessed in my lab with this metabolite itaconate. This is made in macrophages in abundance in response to LPS, for example. We had a paper on this a couple of years ago, several labs now. It's a very potent anti-inflammatory metabolite coming off Krebs cycle of all things. And we've lots of stuff on this to do with macrophages and cytokines in various ways. So of course we tried it in COVID-19. There's the molecule itself. In our paper in 2018, it can protect against sepsis. Here's LPS-induced lethality. In mice, give the mice itaconate, you can prolong life. And then about a year later, a lab working on Zika virus got in touch with me. We gave them our molecule and it protected against Zika lethality. Here's Zika killing mice, give them itaconate, they survive. And that was interesting. Here's a viral-induced sepsis type scenario, I guess. In their paper, I quite like this, the little spray can of itaconate on top of Zika. That could be COVID-19, couldn't it? So of course we've tried it. And we're getting some very interesting results. This is a collaboration, Martin van Hemert in Leiden. This is a lung epithelial model. SARS-CoV-2 infection will kill those cells. You get a cytopathic effect. Give them itaconate, they survive. We can protect those cells from COVID-2 induced death in that cell type. And then literally in the last week, we have a hamster model of COVID-2. This is with our collaborators in Leuven. Lawrence Liesenborg is the key collaborator here. They infect the mice with COVID-2. They did a Nature Medicine paper on this model. It's pretty reproduced, a good indicator of human disease. And they got protection in the histology score, so it improved if we gave them itaconate. This is a complex composite of different markers here. So we see protection with itaconate. Here we see it 4OY. Here's the vehicle. There's 4OY protecting. So it seems to work in vivo. Very early days, but this is yet another approach to limit the inflammatory consequences. In this case, using a metabolite coming off Krebs cycle, where there's emerging evidence of an anti-inflammatory effect. Finally, I'll finish with this. I mean, if dexamethasone works, and let's face it, it does seem to, can we do better? An age-old question. And clearly dexamethasone has taken out some of these pathways. And can we improve on dexamethasone, which we've tried to do for years? And who knows, some of these more novel approaches might lend themselves to that. And one tiny example of how much we don't know. And remember, in my opinion, the hurricane is still raging and we're trying to measure rainfall here. You know, there was a great paper on using AI to interrogate the literature, and bradykinin came out. And my supervisor for my PhD in London discovered bradykinin. He passed away about 25 years ago. Bradykinin came and went as a really important mediator. Who knows, maybe bradykinin might be the key answer to this. And of course, there are bradykinin antagonists in action. Many people, not many, several disagree with the cytokine storm idea, by the way. I think it's overstating it. Maybe it's a bradykinin storm, who knows? So let's keep our minds open to this disease. And I think the big consequence of this may not be COVID-19. The vaccines are looking promising, as we know. It'll be therapies for other situations that are to do with infection or ARDS or sepsis. And who knows, we may get insights that will extend beyond COVID-19 into the future. Thank you very much for listening. I'll finish there, Seamus.
Thank you very much, Garrett. So we'll proceed to our third speaker, Rick Bucala from Yale University. And Rick is going to talk on COVID-19 in the context of autoimmunity. You're still on mute, Rick. Rick, you're on mute.
The anthem of our time, isn't it?
Okay, everyone can hear me. Okay, I'm Rick Bucala at the Yale School of Medicine, and I'd like to give you a sense of the importance of inflammation and also focus on recent information regarding autoimmunity and autoimmune mechanisms that may be having a role in COVID-19 pathogenesis and clinical heterogeneity. I'd like to begin by emphasizing a little bit what we know about the microbial pathogenesis and the immunopathology of this disease. We know it's a respiratory infection by an inhaled virus, produces a pneumonia in 50 to 70% of symptomatic individuals. This pneumonia can be detected by imaging, irrespective of whether it's clinically evident. We also now know that there are 3 phases of stages of the disease, as I've illustrated here. I should note that this scheme we put together from the advanced therapies group at Yale back in March. It really hasn't changed much. The percentages here really have not changed. They're originally based on these very defined clinical epi studies done in cruise ship infected individuals. Gratefully now the overall mortality is much less than 10%, and ICU mortality is less. But having said that, we know that some patients resolve infection. It seems that everyone, to some extent or a great extent, controls viral replication so that viremia reduces in all patients over time, but that some individuals enter this intercritical phase where infection and pathology resolves, or in which pathology is exacerbated. And we've had cases at our institutions where patients have gone home and within 48 to 72 hours are readmitted in extremis, sometimes with rapid death, with progression to ARDS. And then of about 30% of patients in this critical phase will go on into intensive care unit and develop ARDS, shock, and death. With respect to the immune response, as predicted in respiratory mucosal infection, there's an epithelial response involving innate lymphocytes, neutrophils, monocytes, DAMPs, cytokines. There is a strong cytokine response that occurs with and increasingly with respiratory epithelial damage. There's some ideas that perhaps this cytokine, features of cytokine expression, maybe particular cytokines, mediate this intercritical phase and certainly are involved in progression to high morbidity and mortality of shock and death. And there's also still, I think, the lingering question of whether in this intercritical phase, which of course coincides with antibody development, is there antibody-dependent immunopathology that occurs, and antibody-dependent perhaps dissemination through infected cells or through antibody into target tissues to produce the systemic infection that we see in renal cells, in the myocardium, in the pericarditis, which can be detected by sensitive imaging in 25% of individuals. And is antibody involved? And this is important, obviously, with respect to predicting what happens long-term, may be important with respect to immunotherapy and vaccine. There is reports in SARS-1 in a primate model of antibody-dependent enhancement of disease. I'm a reader of that paper who does not think it's particularly significant, but nevertheless, this concern exists with the disease, particularly with respect to clinical heterogeneity. And very importantly, and I don't want to discuss here, from the original Chinese series, autopsy series, we know that there is a severe lymphopenia that can occur in phase 3, which is due almost certainly to excessive antigen presentation by the innate system to the adaptive cells and by excessive cytokine signaling, signal 3 to T cells. And whether this lymphopenia has a role in long-term immunosuppression, failure in sustaining a protective antibody response, and potentially have a role in the potential futility of vaccination. And so clinical consequences, as I've mentioned, the severe ones, ARDS, shock, coagulopathy, which is remarkable, may reflect severe endothelial damage by virus, by immune complexes, and so on, and the additional features I've mentioned. We in rheumatology, as clinicians and translational investigators, are well versed in cytokine expression syndromes. I've tried to summarize them here. Certainly infection, sepsis are drivers of excessive cytokine production. I also am in the group that does not like the term cytokine storm. I think it's an oversimplification of a very complex conditions clinically, and that we are in a state of knowledge now that we can really parse through these conditions and determine which cytokines, which cells are responsible, and potentially which interventions can have a predicted response. And so we know drivers: infection, viruses, influenza, COVID-19, cancer immunotherapy, very well responsive, we know, to anti-IL-6, so a particular cytokine therapy. And in rheumatology, systemic juvenile idiopathic arthritis, which causes macrophage activation syndrome. There's a lot of mention of this in the rheumatology literature with respect to COVID-19. Some of this is driven by somatic mutations in innate cells and responsible, for instance, in the condition hemophagocytic lymphohistiocytosis. Now, from the original series from China, and still true, we know that severe elevations in ferritin, which is a marker of IL-6 and IL-1 driving inflammation in the liver, and IL-6 are predictors of mortality. Numerous other cytokines are associated, and these may not be so much related to their particular function, but to their detectability and their the ability of them to be sustained in plasma. And as I caution everyone, elevation of an inflammatory mediator or a cytokine is not causal. It's an association. It may very well be the case, and those of us who work experimentally know, perhaps the host is not making sufficient amounts of that mediator, and that's why patients suffer from complications, morbidity, and mortality. And so most rheumatologists would argue COVID-19 is not cytokine storm. It is not macrophage activation syndrome. There are shared features. There are shared cytokines, although cytokine expression generally is lower and it's distinct in COVID-19. Cytokine antagonism to date has not been shown to be clinically helpful, although there is still, as in the case of glucocorticoids, who many believed were useful, it's finding the particular patients in clinical circumstances in which particular cytokine interventions may be useful. Okay, now, having said that, there is a distinct COVID-19 inflammatory sequela, which has been described, and that is pediatric multisystem inflammatory syndrome, described originally in the UK, followed up in a study from the French in Paris, which clearly showed an outbreak of a Kawasaki-like disease that occurred in the post-infectious phase in some children with COVID-19. Remember, Kawasaki is a systemic vasculitis characterized by fever, rash, a coronary vasculitis that can lead to aneurysms and lethal myocardial consequences. We have similar cases in the United States, but there are distinct features from Kawasaki, as I've listed here. I don't want to get into the details in time, but it is clearly a post-infectious inflammatory syndrome. I would not say autoimmune because we're not certain if there are particular autoantigens involved, but clearly there's the role of the host response. There's intermediation either of antiviral antibody, maybe particular isotypes or immune complexes. There's something about the immune response that long-term in these individuals that produces this lethal inflammatory condition. It's been proposed, for instance, at our institution, it might be useful in all COVID-19 infected children to measure their antibodies by serology in the post-infectious phase. Perhaps it's isotype, perhaps it's immune complex levels, immune complexes we can't measure, but it clearly has a infectious etiology. Now, what about COVID-19 risk in autoimmunity or in our immunosuppressed patients? And so in many institutions, as in my own, we were quite concerned in our hospital practice, in our hospital and outpatient practice about what do we do with our immunosuppressed patients who are on chronic immunosuppression because of lupus and RA. PPE was limited, but clearly our biologic infusion centers, our infusion nurses, patients, their families, the treating physicians, they all need to be fully protected. So initial information, and this is from the NYU cohort, seems to indicate that in fact, patients with autoimmune conditions or ongoing immunosuppression perhaps are protected from COVID-19 severity. And with the exception of patients that are on modest doses of glucocorticoids, there is no increased risk of hospitalization. And the risk for these individuals is the same as for the general population with COVID-19, age, hypertension, COPD, diabetes. These data were not sufficient to disentangle whether it was the autoimmune condition, which by definition is a type of immunosuppression, or the immunosuppressive therapy, which may be providing relative protection. The same group has also made similar findings in lupus patients, which have potentially a more broader, more systemic type of autoimmunity. But I would also caution that these are early information and that we still need to be cautious and look at individual subjects, particularly as these data play out and as more individuals enter the system. The American College of Rheumatology, for instance, has a global database which is underway for physicians and patients to make these types of inquiries as we follow the course of the infection in our treated patients. Now, the question, and has been asked by my co-panelists, about what about this long tail, the long haulers? Will there be a burden of post-COVID-19 chronic disease? We know from SARS-1 and MERS, of which gratefully there have only been a few thousand cases globally, there's about a 5% incidence of interstitial lung disease. Interstitial lung disease is a post-injurious, post-inflammatory or ongoing inflammatory syndrome. Many of these cases of ILD are associated with anti-synthetase autoantibodies, not a full-blown rheumatologic condition, but an autoantibody of this character. This has not yet been described in the ILD of SARS-1 or MERS, or yet of SARS-2, but I think these circumstances are coming. And what about autoimmune diseases, frank autoimmune diseases? So there are early reports of COVID-19 patients with rheumatoid factors. That's an autoantibody that reacts with the constant regions of other antibodies, anti-nuclear antibodies, which have a general prevalence of about 10 to 12% in the population, and antiphospholipid antibodies, which typically are associated with vascular damage, strokes on both the venous and the arterial side of circulation. But I would caution that having circulating antibodies does not equate with clinical autoimmunity. And so far, in our own health network, I represent the 4th largest health network nationally in the United States. Clinical sequelae have only included post-infection arthralgias, malaise, not surprising, of course, low titer rheumatoid factors, ANAs of unknown significance. And so I think it's important in these cases to really focus on the fundamentals. Biologically, autoantibodies are part of the normal immune repertoire. They're encoded in our germline, generally in low titer. We don't know why we have autoantibodies, but the favorite ideas are they promote innate clearance. They're kind of an innate antibody that may help clear bacterial DNA or immune complexes chronically. And they're well described for the phenomena maybe 100 years in chronic unresolving infections, syphilis, tuberculosis, endocarditis, malaria. They are a feature of infection, of severe infection. And autoantibodies, we also know, importantly presage the development of autoimmune disease. If you have an ANA, or an anti-rheumatoid factor, CCP, your risk of developing frank clinical autoimmunity is about 30% over about a 5 to 10 year period. It's probably related to mucosal injury and autoantibodies that are triggered at mucosal sites, which may be telling us something important if we see this in COVID-19. But very importantly, and let me end by emphasizing, autoantibodies as detected do not equate with autoimmune disease. We have not yet seen that, but because of the severe mucosal infection that has occurred, and because millions now in the global population across a very broad, diverse background of genetic susceptibility, frank autoimmune disease may very well be coming, including new to be defined autoimmune syndrome. And so the final word, I think, here for all of us in medical practice, particularly in primary care and on the front lines, is vigilance. Vigilance in the post-COVID-19 patient, And this must include, as we are doing in our institution, continuity clinics for patients who have COVID-19, both by infectious disease, pulmonary services. We are initiating questionnaires of patients, for instance. Do you have first-degree relatives with arthritis, with a rheumatic disease, with autoimmunity? These may all portend importantly for the development of autoimmune disease in the post-COVID-19 era in patients. Thank you.
Thanks very much, Rick. We'll move on to our final speaker. I'd like to welcome Professor Timothy Tellinghausen. And Tim is head of virology at Roche. Tim, you're very welcome.
Hope you can see my screen.
Yeah.
So in terms of assessing the immunity to COVID-19, it's a complex subject, and I thought I would approach this around the question of where might immunity come from. And inside that subject, there are three areas of active debate in the literature that I thought were quite fascinating and I'd like to share today. And really those are, one, immunity from infection with related viruses. The second is immunity from unrelated viral vaccine cross-protection. And the third is immunity from infections with SARS-CoV-2 and how long that might last. So I'll give you my opinion on these issues from my interpretation of the literature. Please be aware that my opinion may not be representative of reality, and the scientific progress in this field is rapidly evolving, so this may change over time. But let's dig in. So what can we infer about COVID-19 immunity from related viral infections? So 4 coronaviruses are endemic human pathogens. These are seasonal respiratory tract infections of global distribution. And these viruses do not provide long-lasting immunity to reinfection. There have been several challenge studies over the last 50 or so years showing that reinfection is possible after as short as a year, although with symptoms that are milder and with lower titers in those infections after 1 year. But if you challenge after a couple of years, you get basically back to normal infections with normal titer and normal pathology. So that tells you a little bit about coronaviruses and that they're unlikely to establish long-term immunity. So back at the beginning of coronavirus, someone asked me what I thought about the potential of developing immunity from endemic coronaviruses. And I told them I didn't think that was going to be a likely path to success based on the low sequence identity between the spike protein of these different viruses, plus the fact that they don't induce immunity to homologous challenges, that challenging them with a very different virus would be unlikely to be successful. But surprisingly, the literature has highlighted that there's a lot of really interesting things happening here. Probably the most exciting thing is that some number of studies looking at COVID-naive patients, specifically children, identifies some fairly robust B and T cell responses to SARS-CoV-2. These are patients who've never been infected before, but they can recognize peptides from SARS-CoV-2, suggesting that this came from a previous endemic coronavirus infection. You see this in adults as well, although there the B cell response is much weaker and it's a more predominantly T cell response. The challenge, that sounds like it'd be a very good thing, but the spectrum of epitopes that you see recognized from natural are different from natural infections. And some of these are unlikely to be neutralizing, but there still are some things in there that look like they might be capable of neutralizing virus. So that's an interesting observation. And when you look at the numbers, something like 50% of children have these sort of antibodies or sort of antibodies that recognize SARS-CoV-2. The challenge is that when you look at what happens in pediatric patients, there are literally millions of infections. So this tells you that, it's maybe not protective. It's hard to say because we haven't really done a lot of sampling in that space. But we do know that children often show less severe disease. So the question is, could existing naive responses from proximal endemic coronavirus infections provide some protection to COVID disease, like we saw in these challenge studies? And there's a lot of suggestion that maybe some endemic viral infections produce this sort of protective effect. If you look in sub-Saharan Africa, there's been a relatively mild incidence of COVID-19 disease there. And a number of serological studies suggest that endemic coronavirus infections there are really common. Over a span of 5 or 6 years of a study, you'll have people infected with the same virus 3 or 4 times, suggesting that there's maybe perhaps much broader immunity there. So I think this is an area to watch develop. I think it's unlikely we're going to see sterilizing protection from endemic coronaviruses, but I think perhaps we'll see some disease limiting effects. So jumping to the second area, vaccine cross-reactivity based COVID-19 protection. Can heterologous vaccines that we all received in childhood protect us from SARS-CoV-2? And I think this is based on a number of publications and literature of small stretches of peptides common between largely measles and rubella, envelope proteins and SARS-2 spike, as well as some intracellular proteins. These have been proposed to potentially provide protection. Even in the best case, they look like there's not a lot of these potential epitopes, and they're not likely to produce what we think is required for immunity, which would be a robust polyvalent immune response. In fact, a recent large-scale analysis found no shared T or B cell epitopes between SARS-CoV-2 and every vaccine-preventable pathogen, suggesting that this is not probably a likely path. And I think that my opinion is that this is probably not a relevant way to get specific protection from SARS-CoV-2. I want to highlight that this is different from the use of vaccines as more general immune stimulators. Taking a safe vaccine and using it to ramp up the immune response, that's quite a different paradigm. And there's ongoing studies for BCG and others, and I think that's a viable path. But I think just getting protection from something like the MMR is unlikely. And we see many, many patients in places like the United States where MMR prevalence is very good, being infected. So I don't think there's probably a lot of evidence for that being a viable path. But it continues on in the literature that there's some potential protection. And then the last area really is the big one. What about natural COVID-19 infections and the development of immunity? So we know that there is probably some protective immunity. I mean, reinfection seems to be a very rare thing, suggesting that some protection exists during the time frame of the current pandemic we're living through. There's something like 55 million cases, and you measure the number of reinfections in the thousands, suggesting that there's probably pretty good immunity, at least for the last year or so. So the real question, though, is how long is that going to last? Is this going to be like those endemic coronaviruses I told you about back on that second slide there, where immunity fades quickly? There's a lot of publications and literature really highlighting the significant variability in antibody durability from many different studies. And it's very hard to make any sense of this. Some say antibodies last for a few months, some say they last for 8 months. It's really hard to really get anything from this. But I think a quite large and nice long study was just released as a preprint from the La Jolla Institute of Immunology. And I think it's a fascinating study. It shows that S antibodies really last for the entire time that they've been measuring, 6 to 8 months for the time frame of the study, without any significant fading. And B and T cell memory response as well is quite robust and durable. And this includes patients from a range of various clinical outcomes. So it's not just people with asymptomatic infections, but it also includes severe cases and other backgrounds of clinical samples. So I think this tells you that you're going to get some protective response that's going to last for months to a year maybe. If we look at what we know from SARS, from back in the days when that virus was running amok, people have been following those patients for a long time. And then we know that patients lose their antibodies to SARS over a few years, 2 or 3 years, that fades. And the B cell memories fade as well, typically over 4 or 5 years, something like that. T cell memory seems to persist. But again, it has a different immunodominance and it recognizes different epitopes than you see in a normal early infection. So it's unclear how protective they would really be to a challenge with SARS again. Let's hope that we don't see SARS ever coming back again to challenge those patients. But for now, it seems like in patients, at least the antibody-mediated responses fade. And a number of really exciting papers in Cell just recently suggest that SARS-CoV-2 infection actively blocks B cell memory responses by basically inhibiting the function of T follicular helper cells, which are important in establishing immune memory from B cells. But it seems the memory T cells are not impacted, at least in the short term. But there's a lot of evidence from clinical infections that there's a lot of lymphopenia and other things that might impact the ability of the body to establish an immunological reservoir capable of recognizing virus really well in future challenges. So my feeling is that B cell immunity will fade over a few years, much like we saw in SARS. And the altered T cell memory may or may not be protective. I'm thinking probably it will not be, based on what we've seen in our own internal studies and looking at peptide recognition distribution. So just in conclusions, very quickly, I think, infection with endemic coronaviruses doesn't seem like it's going to provide really great protection. In some populations, you might see it will limit disease. Maybe children and some populations with very regular infections might be better off than people who don't regularly see coronaviruses. It's a developing area that we want to watch. Vaccines to other pathogens, I don't think are going to provide a lot of protection. There's just not antigenic peptide overlap that makes this seem like a logical choice. Again, I highlight that things using vaccines that are safe as immune modulatory agents is a different story, and that may be a viable path. And then COVID infections, I think, will provide some immune protection of natural immunity, but I don't expect it to last, which is unfortunate. I think reinfection right now seems to be rare, so we'll get some protection in the short term, but I don't know of any coronavirus that induces long-term protective immunity. But I think the silver lining in this is that I think, despite all my predictions of sounding very negative, I think that vaccines are a very different situation to a natural infection. You don't have the virus messing with the immune response. So I think a vaccine delivered through, into the blood, for example, versus through the mucosal surface may be a very different paradigm. And we may find that vaccination might produce a durable response. And I certainly I hope so, because I'd love to leave my house again. So with that, I'll end and pass back to the moderator.
Thanks very much, Tim. So just encourage viewers, online viewers, to ask questions via the chat format or the chat button down below. So I suppose get the ball rolling. So why, if I can, if there's an index case of COVID in a household, why do up to 50% of first-degree relatives of that household not get COVID infection, certainly symptomatic COVID infection? And it's a very broad question, but what is the basis for that? Does anybody want to take that up or give a... Luke, do you want to say a few words on that? Or
Yeah,
why
I think the word is good luck, Seamus, in a way, This is a very statistical business, remember. It's very random almost, and I think dose of virus, wrong place at the wrong time, that kind of thing is part of this, Overall health status, of course, is a factor. So there's loads of reasons. I mean, friends of mine say, oh, I had the virus and didn't infect anybody in my household. And I try and explain to them along those lines, I guess, it wouldn't be genetic necessarily if they're in the one family, So I think it's just randomness, really.
Okay, all right.
My
answer anyway. I don't know what other people think.
So Colin?
No, Rick wants to come in. Go ahead.
Oh, Rick, yep.
If I can comment, Seamus, my understanding is that having an infected family member conveys the greatest risk of being infected. Maybe it's only 50%, but in terms of social settings, it is the greatest risk.
Okay. Apart from religious conventions in the Deep South.
Yeah, I was going to highlight that.
Or yodeling in Switzerland.
Exactly. I think there's some really fascinating things. If you take the worst case scenario type situations, these choir groups and the very famous Swiss Alphorn and yodeling competition, in those cases, which you have people behaving in the worst possible way in terms of viral transmission, we have numbers there of some of these that are 80 and 90% transmission, right? So I think in that context, it doesn't look like there's a whole lot of protection. In a household where there's different rooms and air currents and people don't stay in the same room, or some of these restaurant studies that have been published out of China, it seems proximity is the big driver, or sitting near an air conditioner or a ventilation system. But yeah, I think it comes down to exposure and luck. But I don't think it's related necessarily to a natural genetic distribution in humans that's really common. I mean, we know those exist, but they don't seem like they're really common events.
Okay, so questions coming through here in relation to seasonality and vitamin D. What's the panel view of, should we all be eating like Smarties vitamin D, or what's the view? Garrett, do you want to get the ball rolling on this?
So, only to say there's a lot of hand-waving stuff out there, and that the real indicator is how little we understand seasonality.
Okay.
I think it's an intriguing area where the amount of science in terms of understanding what's going on is pretty trivial.
Okay.
I mean,
Seamus,
I
can
think,
I put-
in my view, the good bit is we know a fair bit about vitamin D in the immune system, remember. There's lots of studies going back, you know. So there's a rationale, there's a scientific rationale, but you end up with the causation, correlation, association business again, you know. So it's very hard to pin down. I wouldn't be that, well, some of it's convincing actually, but overall the jury's out in my view. Certainly just going out in the sunshine isn't going to stop you getting COVID-19, let's put it that way, you know.
Okay.
I would add that, you know, I think this is what Professor O'Neill is saying, that the epidemiology studies for vitamin D seem quite, in different diseases, infectious immune, seem quite robust and well done, but they're association studies.
Lots of covariates.
Equally, vitamin D supplementation has not been shown to be useful, to my understanding, in anything except for frank vitamin D deficiency.
Okay. All right. So Tim, can we come back to you again with regard to susceptibility or protection, the children aspect, the pediatric or the children aspect? Why is that? What's so special about children and their immune system? Or is it more kind of go back to pregnancy and passive transmission during pregnancy, or is it post childbirth?
I think it's post childbirth. My feeling is that children don't have necessarily as a robust immune response as adult, but they do constantly get exposed to these sort of anyone who has children knows they are regularly exposed to sort of these constant upper respiratory infections, and among those you see a lot of coronaviruses. This is an area that we've worked at Roche for many years. Spinning off from influenza, but it also includes RSV and a number of other respiratory infections, largely focused on pediatrics. So we've put a lot of effort into this space over the years. And I would just say that the challenge, I think, is that upper respiratory infections in children just do not produce really robust immune responses. So they get infected. It's when they move to the lower respiratory tract that you start to see a ramping up of the immune response. And that's where you get, I think, most of the protection. So, I think these endemic viruses give you a little bit of a boost. And then as you keep getting them, as they come back every season or every year, you get sort of very similar to a vaccine boost, prime, or prime boost, I should say. So I think you get this development of antibodies that exist for endemic coronaviruses, and there may be some protection from that. Again, this is very, it's speculative stuff based on some new things in the literature. So, I wouldn't advise any physicians to change their treatment strategy based on my suggestions, but I think it's an intriguing area of science that, that this exposure to other viruses may have some protective effects, at least in terms of disease severity.
Okay.
Seamus, can we just pick up one of the questions, because it's a question I wanted to ask as well, so from the chat, which is about any specific features of this virus that immunomodulatory. Could we guess that this was coming if we have a further coronavirus? Are we going to guess which are the bad ones? Does anybody want to weigh in on that?
I certainly can. We've been looking extensively at coronaviruses in a very systems biology sort of way, including many coronaviruses, because big companies, we have a lot of research funding to do that kind of stuff. So we've been really actively looking at coronaviruses, trying to define what makes a bad coronavirus detectable relative to a good coronavirus. And it's not as easy as it sounds. I think what we've really settled in on is thinking that and I can't tell you anything about what we're actually really doing in terms of early discovery, but we're focused very much on making pan-coronaviral inhibitors. So we're certainly focused on delivering things to the clinic now because there's a very high unmet need for the current pandemic. But a lot of our discovery efforts are focused on inhibiting broader families or subfamilies of viruses and trying to develop an inhibitor that might work on many different coronaviruses, rather than trying to define, which bat virus is going to, recombine with what other virus and then become the next pandemic, because it's very hard to predict that. We haven't been able to identify what that signature of a bad coronavirus looks like. So we're trying to develop broad inhibitors.
Okay, thank you.
The challenge, Colin, is to look at the number of proteins the virus encodes and what they target. And this has been going on for years with vaccinia. It began with vaccinia, of course, all those years ago. So it's very difficult because each viral protein might have 30 host factors it interacts with even, you just don't know. But one thing, a very interesting paper just came out, I think it was in Nature, on why, as you would all agree, I imagine, it's asymptomatic spread is one of the most pernicious parts of this virus. So, And there was a paper showing it's very good at suppressing NLRP3 in the upper airways. And that's why there's no symptoms. You know what I mean? And yet it still provokes type 1 interferon. So there's something about this virus that it can allow itself to transmit from asymptomatic subjects, if you will, which is very different to SARS and MERS, you know. And it's something to do with immunomodulation that allows it to do that. And that, as I say, that's probably the most pernicious part of the whole story so far, because it keeps spreading now. You know, you can't get rid of it too easily if that's the case.
And actually that leads to another set of questions in the chat, which is about whether the interferon pathway is going to be tractable to deal with this disease. Does anybody want to
there's good data coming out with inhaled interferon now, isn't there, for example, I think. I mean, we may finally see a blockbuster drug. Would you agree, Timothy, with type 1 interferon? You never know. I mean, and Garrett mentioned those great genetic studies are fantastic by Casanova's lab. I mean, that really further highlight. I mean, we knew about this pathway for donkey's years anyway, but there's something going on with COVID here for definite. I think that's going to be another potential therapeutic that will actually might work on help patients.
No, I agree. Absolutely, I think.
Can I just follow the therapeutic angle then? In relation to the timing aspect, and Garrett, you highlighted this. We've got the N=1 Donald Trump trial, where he was given the therapeutic cocktail, you know, on the first sniffle, or the drizzle, the cytokine drizzle. So can you just allude or highlight or pull out this timing aspect?
Well, it's always nice to start with an anecdote that involves our ex-president. So, as you know, he is actually. Let me tell you. What was interesting about Trump is that he was clearly severely ill. He would not have got steroids if he wasn't. And the interesting thing was that this obese person bounced back to be remarkably active in a very short period of time. And as you say, he got the Regeneron drug very early. So, you know, whether that anecdote means anything or nothing is neither here nor there. The issue is that we know that the immune response initially is beneficial. And later the immune response runs out of control and it's adverse. So it makes absolute sense that the timing of intervention, no matter what the intervention is going to be critical. And we've already had a few hints of that. You know, the Lilly trial that was negative came a bit late, and people wonder about remdesivir indeed as well. But be that as it may, that's where I think gathering the science on the evolution of the immune response is really important. So what we know from query studies is that if you look at the extreme phenotype, people sick in ICU, there were 4 distinct immune responses, including people who didn't develop antibodies at all. So what we need to do is go earlier in the disease process, look at the distinct evolution of the immune response, and then determine whether that gives us indication as to timing of intervention and the type of intervention. And I'll sort of highlight that by saying, if you take something, and Luke alluded to all the studies of LPS, there are 1,000 drugs that if you give them before you give the animal a slug of LPS, prevent the evolution of ARDS. Well, here we're actually potentially in a useful situation. In those animal studies, if you give it once the disease is underway, these drugs don't work at all. And we've tried these drugs in patients with established sepsis, and they don't work. But here, we have a much earlier stage of evolution of the disease, a bit analogous to the opportunity to intervene before the animal develops ARDS. And it could well be that many of those drugs that didn't work in the other situation could now become effective. So, you know, we believe we've a big interest in lipids as modulators of viral response and of the inflammatory response. We think this is a pretty underdeveloped area, given how potent these diverse lipids can be, both at the viral end, but also in terms of the diverse aspects of the immune response. And many of these interfere upstream, for example, of the type 1 interferon pathway, upstream of inflammasome activation. So I think there's a lot of biology to understand here that we just don't have at the moment.
So I'm going to So Seamas, probably we need to wrap up now. There's been lots of other questions, been really interesting, particularly about the thrombotic cascade and how that gets involved. But I'd just like to conclude by saying the COVID enigma continues to be a COVID enigma. I think that comes loud and strong from all of the speakers, but amazing science has been brought to bear, you know, in just those 8 to 9 months has really been astounding, and we hope we'll benefit much more broadly from that. I would just add on my own part, thanks to all of the speakers, but I think in science, you also need a bit of luck, don't you? You also need to get the timing right, and you need to intervene right. And maybe we've had a bit of luck from the vaccine side. Let's see how lucky we can get with some of the other interventions. But as Gary Player says, the more I practice, the luckier I get. So the more science we do, the more likely we are to be successful. So thank you to everyone. Thank you all of you who've stayed at different times to join us today. To Seamas, my co-chair, and we wish you all to stay safe for the future and look out for future episodes of emerging issues in translational medicine from the Association of Physicians. Thank you all.